proteins are made or polypeptides are produced. Okay, DNA is transcribed into RNA, messenger RNA, then the messenger RNA is then translated into the polypeptide or whatever product you want to produce. Okay, so now we got to figure out how these genes are expressed. Okay, how are they turned on and off? So this is gene expression. It's been well studied in prokaryotes where they used what are called operons. Okay, so we're going to look at two different operon systems today. Okay, one is first of all, let's define operon. Operon is a unit of DNA. Okay, so a strand of DNA which is going to produce a single messenger RNA. Okay, actually we can expand it. It can produce different messenger RNAs to get a single product. All right, so it's a kind of a system, maybe producing genes for a metabolic pathway. All right. Okay, so this should be somewhat familiar to you. All right, so we have the promoter region. Remember, that's -10 or -35 sequence upstream, where the messenger or the RNA polymerase is going to attach. Okay, when it attaches there, then it can start reading down. When it hits that that start codon, which will produce AUG, it's going to start producing that messenger RNA. Okay, so here's the gene. In this case, this is we're going to look at the trip operon. Okay, the TRP, or the tryptophan gene, is right here. Okay, so it'll produce messenger RNA, which will then produce this amino acid tryptophan. All right. Okay, so that's what happens there. Okay, that's normal. All right. So within that promoter, there's a thing called an operator that controls the access of the RNA polymerase. Okay, so in this particular system, okay, trip operon, okay, it's normally on, all right? Okay, but upstream a ways, there's regulatory genes which operate, okay? Okay, so there's a promoter for that, of course. So there's this gene produces a messenger RNA, and this messenger RNA will produce this repressor protein in an inactive state. Okay, so it produces this protein, which of course has a characteristic shape, okay, but it's not active at this point, all right? Okay, so what happens though, and you guys remember negative feedback, okay? So negative feedback means high levels will turn substances off, okay? So in this case, this system is normally on, okay? This trip operon normally on, so you're producing this tryptophan. When this hits high levels, the tryptophan can go over and interact with this repressor protein. Remember that allosteric sites? It can bind on here, and when it does that, it activates it. And once this is activated, okay, then it can go here, and it locks onto this operator site, which controls the access of the RNA polymerase. Right? So when you have high levels of tryptophan, okay, it'll go on and it'll activate this repressor protein. And when you activate a repressor, okay, it will repress the production of the trip gene. Okay, so then of course, after a while, your levels of tryptophan will go down. And of course, when that happens, okay, it'll no longer bind there, okay, and the repressor will no longer work, and then you can continue production of it. Okay, so a great example of negative feedback here. Okay, increased tryptophan will bind inactive repressor, therefore it activates that repressor and it will stop the production. Okay, so this repressor protein will just lock on there, and then the RNA polymerase can't get a hold of there, and so it'll stop production. All right, so that's the all right, class. Now here's the question: what is the #1 sugar? Okay, probably the primary sugar that's being used for energy. Well, if you said lactose, sucrose, maltose, you would be wrong. You'd have to say glucose is the primary one, which most organisms use to break down. Right? So glucose is normally used. So this is another operon system where the system is normally turned off, so it's repressed. Okay, then you can turn it on. So this is called the Lac Operon system. Again, it's well studied in prokaryotes. Okay, so here's how it works. All right, so you got the regulatory gene upstream from the operon system for this lactose breakdown enzymes. Okay, so here's the regulatory gene right up here. You got the promoter. Here's the gene produces messenger RNA, then it produces an active repressive protein. Okay, so this is active. So this active protein can go in and lock up the operator over here. Okay, of course we said the operator, it controls access of the RNA polymerase. Okay, so when this is locked in place here, RNA polymerase cannot bind. Okay, so this whole process shuts down. So it's normally just shut off. Okay, so as long as you have plenty of glucose, okay, it's going to, this system's going to not go, all right? But what happens is it somehow gets, the repressor gets turned off, okay, so what does that is if you get a real large increase of lactose, okay, increase in lactose here, will bind the repressor. So if you get a lot of lactose, okay, build up, so that means that sugar levels, glucose levels are going down, okay, so lactose is probably going to increase, okay, or that's the food source, okay. So what happens there, the increased lactose, well, just like before, it's going to, there's an allosteric site on that repressor, okay, which is then going to be locked in, and it's going to deactivate this repressor. So no longer do you have this repressor protein here, okay, so it opens up. So now the RNA polymerase can come into the system, okay, and produce messenger RNA, produce these enzymes which will break down lactose, okay. So then, of course, when lactose levels go down, decrease in lactose, okay, that means this repressor then will then activate again, and then it can lock up. Okay, so again, it's negative feedback. Increased lactose will bind the repressor, which will inactivate it. Okay, but low levels of lactose will then turn it back on. Okay, so again, negative feedback. Okay, again, operon system. Okay, in this case, it's off until you turn it on. Okay, whereas with the trip operon, just the opposite. It was on until you turned it off.